This video presents an overview of the SODAR data management system. It is being developed by Core Unit Bioinformatics at the Berlin Institute of Health. SODAR stands for the System for Omics Data Access and Retrieval. It is specialized software for managing study data in various omics research projects. SODAR provides biomedical scientists with a single point of access to data in their projects, along with linking out to external resources and systems. The main features of SODAR include project-based access control and data encapsulation, modeling study design metadata, large-scale data storage, linking files to metadata, validation of file uploads, and various tools for aiding in data management. SODAR offers a user-friendly web-based graphical user interface for scientists. REST APIs are also provided for programmatic access from scripts and other software. SODAR has been designed to support the FAIR principles: findability, accessibility, interoperability, and reuse. In the SODAR data workflow, sample sheets link together researchers, study design, and data storage. Sample sheets are used to model the study and store metadata for samples, processes, and materials. Studies are defined using the ISATAB standard and displayed to the user as spreadsheet-style tables. Through sample sheets, the user can access files, shortcuts, and external resources. Large-scale sample data is stored in a distributed file system built on the iREDS open-source data management software. The sample sheets link out to iREDS collections related to a specific study, assay, or sample. SODAR can be used to store all types of files relevant to your studies, from large binary alignment map files to results and reports. Uploading sample data is done using landing zones. Landing zones are user and assay-specific temporary file areas which grant the user full read and write access for uploading and manipulating their files. Uploads are then validated for integrity and moved into the sample data repository in an automated process. Afterwards, they can be accessed in the sample sheets application. The sample data repository for each project is read-only and immutable. In case of a mistake when uploading, the user can submit a deletion request, which needs to be accepted by the project owner or a user with similar permissions. SODAR supports different methods of authentication, from local users to multiple LDAP servers. Data is encapsulated into projects with several access levels which can be assigned to users as project roles. The same user permissions are shared between the projects in the web UI, REST APIs, and iREDS file storage. User-specific access tokens can be generated for REST API use. It is possible to limit their expiry date and revoke existing tokens. UUIDs and permanent URLs are provided for each relevant object in the system. The SODAR UI provides site-wide search functionality, which covers everything from project metadata to alternative donor IDs and files in iREDS. For easy access to iREDS, SODAR automatically generates required iREDS configuration files for each user. The iREDS system used by SODAR also comprises a WebDAV server for web access and network drive mounting. For complex data transfer operations, SODAR features an iREDS transaction engine with rollback capabilities to ensure data integrity. The timeline application in SODAR is used for advanced logging and audit trails. Timeline stores a log of actions on project objects with a UI to track modifications. Information is maintained even if related objects are deleted. SODAR automatically generates shortcuts to relevant study files in iRODS. This includes the most recent BAM and VCF files for germline and cancer studies, as well as raw data and MaxQuant links for proteomics projects. For use with the integrated genomics viewer from Broad Institute, SODAR can generate and open session files for selected data. Merging files and sessions happens with just one click. SODAR also allows creation and management of track hubs for integrating with the UCSC Genome browser. SODAR enables editing sample sheets directly in its UI. Sample sheets can be created from templates or imported from existing ISATAB files. Cells can be edited in the UI in a similar fashion to a spreadsheet application. For each column, the accepted format, value choices, value ranges, and related options can be configured in order to match the study's requirements. Adding and removing rows is also supported for study and essay tables. For each edit, SODAR validates the changes using the Altamisa parser also developed at Core Unit Bioinformatics. SODAR supports ontology term lookup for numerous common ontologies. These ontologies can be uploaded into SODAR in the OBO format for fast and reliable ontology term queries. Multiple ontology terms can be linked to a single cell. When editing sample sheets, old sheet versions are stored as backup. These versions can be compared and restored in case of mistakes. To use the sample sheets outside of SODAR, they can be exported in the ISATAB TSV format or more simple spreadsheet tables. SODAR is based on open source technology. It is built using Python, the Django web framework, and Vue.js. SODAR has been in active development for several years. It is currently in use in a large number of projects at BIH. Development is ongoing and new features are constantly introduced. The source code for SODAR and associated projects has been made publicly available at github.com/bihealth. The SODAR user manual can be found on readthedocs.io. Reusable components of the SODAR system have been bundled into a separate package called SODAR Core. This package can be used to build web-based tools sharing the SODAR framework of project and access control, application development, and general user interfaces. Thank you for your interest in SODAR. See our website for more information on the work of Core Unit Bioinformatics.